Why do cells put specific RNAs outside? What are they doing? How do they get there? Are they different in disease? But it's a very different model of how a cell is organized, how the body is set up, right? And so, there's a lot of mechanistic questions that's what I like thinking about. But the kind of really cool thing I think is that because we are on the outside of a cell, this is all where the drugs work. This is all where the diagnostics work, right? So when you make an antibody, a monoclonal antibody to target something, traditionally you'd think, well, I got to target a protein. That's the only stuff that can access, right? If I'm going to make a CAR T cell, it's got to target a protein that's going to be on the surface of the cell. So one of the things that we're very excited to consider, right, this is the proposition that we're bringing to the field, is maybe there is RNA out there. Maybe they are accessible to therapeutic targeting, to diagnostic detection. And what would that mean? Why are cells doing it? And while we're figuring out why, we're also thinking a lot about, well, we know it's there. So what if we start targeting it, right? Is there a way that we can actually build new drugs that target this biology that might be better anti-cancer agents, or that might explain why people develop autoimmune diseases, these kinds of things? So there's a lot of very deep biological questions that we've been asking, which will take many, many years to figure out. But there's also a lot of empirical work that we've been doing that's trying to bridge it to the clinic faster than I would have expected, but is actually quite exciting.